To begin the process of gluconeogenesis, pyruvate first enters the mitochondria.
In the matrix of the mitochondria, pyruvate carboxylase converts pyruvate into oxaloacetate. It does this by using biotin and ATP to attach a carbon to pyruvate. Oxaloacetate.
Now that I am an oxaloacetate, I am unable to leave the mitochondrial matrix of the cell.
In order for oxaloacetate to leave the matrix of the mitochondria, a malate dehydrogenase will convert oxaloacetate into malate. That is accomplished by using a high-energy electron transport molecule, NADH, to reduce oxaloacetate, convert it into malate, and disguise it so that it is able to leave the matrix of the mitochondria. Now that the malate has left the mitochondrial matrix, we are now in the cytosol, where a cytosolic malate dehydrogenase will now oxidize the malate into oxaloacetate.
Oxaloacetate gets converted to phosphoenolpyruvate using phosphoenolpyruvate carboxykinase.
Phosphoenolpyruvate carboxykinase will use another high-energy molecule, GTP, to phosphorylate the oxaloacetate and then remove a carbon to convert oxaloacetate into phosphoenolpyruvate. The next following steps in gluconeogenesis are simply a reverse reaction of the pathway of glycolysis. So in this step, phosphoenolpyruvate will be converted by enolase into 2-phosphoglycerate. This is done by a hydration reaction which converts to 2-phosphoglycerate.
2-phosphoglycerate gets converted to 3-phosphoglycerate using the enzyme phosphoglycerate mutase.
Phosphoglycerate mutase accomplishes this by attaching a new phosphate to the 3' carbon and removing the phosphate from the 2' carbon.
3-phosphoglycerate gets converted to 1,3-bisphosphoglycerate using the enzyme phosphoglycerate kinase.
Phosphoglycerate kinase does the reverse ATP synthesis reaction by using a high-energy ATP to phosphorylate 3-phosphoglycerate and attach a second phosphate, making 1,3-bisphosphoglycerate.
1,3-bisphosphoglycerate gets converted to glyceraldehyde 3-phosphate via the enzyme glyceraldehyde 3-phosphate dehydrogenase.
Glyceraldehyde 3-phosphate dehydrogenase will use NADH to reduce the 1,3-bisphosphoglycerate and will remove a phosphate to make glyceraldehyde 3-phosphate. The next step of gluconeogenesis is an isomerization reaction converting glyceraldehyde 3-phosphate into dihydroxyacetone phosphate. This reaction is carried out by triose phosphate isomerase, which will convert glyceraldehyde 3-phosphate
into dihydroxyacetone phosphate.
The next step is carried out by the enzyme aldolase, which will join another glyceraldehyde 3-phosphate with the dihydroxyacetone phosphate,
forming fructose 1,6-bisphosphate.
The next step of gluconeogenesis is the conversion of fructose 1,6-bisphosphate into fructose 6-phosphate, using an enzyme that is unique to gluconeogenesis, fructose 1,6-bisphosphatase. This enzyme will use a hydrolysis reaction to cleave the phosphate, forming fructose 6-phosphate.
Fructose 6-phosphate, a ketose, is converted to glucose 6-phosphate via the enzyme phosphoglucose isomerase.
Ketose, aldose. In the final step of gluconeogenesis, glucose 6-phosphate is converted to glucose by the enzyme glucose 6-phosphatase, which uses another hydrolysis reaction to cleave phosphate.
Now I'm glucose and I can leave the cell and go to other tissues and provide energy.